This method produces highly reproducible hepatocyte-like cells in a 96-well format by using semi-automated platforms.
The main advantage of this technique is that it's rapid, cost-effective with reduced variation, and allows the mass production of 96-well plates of hepatocyte-like cells.
Prepare a 5 microgram per milliliter solution of thawed laminin 521 by diluting it in ice-cold 1 times Delbecos phosphate buffered saline, or DPBS, with calcium magnesium. Use an automatic liquid handling dispenser with a standard tube dispensing cassette to add 50 microliters of the laminin 521 solution per well of a 96-well plate. Incubate the laminin 521 coated plates. After incubation, aspirate the laminin 521 solution off the coated surface using an 8-channel aspiration adapter and avoid any contact with the coated surface. Immediately add 50 microliters per well of M-Teaser-1 medium, supplemented with 10 micromolar of Rho-associated kinase, or ROCK inhibitor Y27632. Keep the plate in the incubator until cell seeding at 37 degrees Celsius and 5% CO2. From a cell suspension of 3 times 10 to the 5th cells per milliliter in M-Teaser-1 medium supplemented with 10 micromolar ROCK inhibitor Y27632. Add 50 microliters of the cell suspension onto the plate. After seeding, return the plates to the cell incubator at 37 degrees Celsius, 5% CO2, for 24 hours to allow